Assalamualaikum. My name is ADIL bin Muhammad ANUAR. My matric number is 23DBT201F1016. So now I'm going to do a DNA extraction biotechnics virtual lab. So in this experiment, we need to isolate some DNA from a human test subject, and why we need human DNA. So this is example. So scientists isolate DNA for a variety of reasons, some of which include genetic testing, body identification, and analysis of forensic evidence. So, DNA extraction is a routine procedure used to isolate DNA from the nucleus of cells. So, basically, when an isocold alcohol is added to a solution of DNA, the DNA precipitates out of solution. If there is enough DNA in the solution, we will see a singly white mass. So DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of that process because the DNA first needs to be purified away from proteins and other cellular contaminants. We need cells because that is where the DNA is. Inside almost every cell in our bodies is a nucleus. So inside each nucleus, about 2 meters of DNA. DNA. So first, we need to collect some cells from our test subject. So we will take a cheek cell for the test subject. So here is cheek cell. It's taken away. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we are after. So let's get started. So this step we will follow to purify DNA from a cheek swab are shown below. So there are 4 steps that necessary for DNA extraction. First, we will collect cheek cells, burst cells open to release DNA, and separate DNA from proteins and debris. And lastly, we will isolate concentrated DNA. So here we can see the material and equipment we will use to purify DNA from cheek cell. Water bath, microcentrifuge, buccal swab, micropipettes, and sample tubes. And we have 5 solutions. So the first step is I will drag the swab over to the inside of the test subject mouth to collect a sample. So here we will get the cheek cell. We will place the swab into an appended tube. So we will look closely at the swab. We will take note that it is covered with hundreds of tiny cheek cells. Inside each cheek cell is a nucleus. So inside the nucleus is DNA. So the end of the swab must be cut off so we will able to close the tube. Using the micropipette, we will add some lysis solution to the tube. And lysis is a Greek word that means to separate. So means we will separate the swab, the chick cell. Okay. So we will place the tube into the warm water bath. Yes. So the lysis solution we just added before contains two important ingredients, that is detergent and an enzyme called proteinase K. So the detergent disrupts the cell membrane and nuclear envelope, causing the cells to burst open and release their DNA. So the DNA is still wrapped very tightly around proteins called histones, and the proteinase K cuts apart the histones to free the DNA. So now the cells have stayed in the warm water bath for long enough for the DNA to be free from the cell, and we will have removed the swab from the tube, and we will add some concentrated salt solution to the tube. So the salt causes proteins and other cellular debris to clump together. Now we will place the tube into the centrifuge. Close the In order to balance the centrifuge, a tube containing water is placed opposite the tube. We will click the lid to close the centrifuge and turn it on. So, after the centrifuge step, inside the centrifuge, the tube spins around at high speed. The heavy clumps of protein and cellular debris to sink to the bottom of the tube, while the strands of DNA remain disrupted through the liquid. So there will be a precipitation of protein and debris inside the DNA and liquid. So we'll use the micropipette to carefully remove the top liquid, which contains DNA, and place it into a clean tube. The proteins and other cellular debris stay behind. So lastly, we will isolate concentrated DNA. We will use some isopropyl alcohol to the tube. So inverting the tube several times mixes the isopropyl alcohol into our DNA solution. Because the DNA is not soluble, does not stay dissolved in isopropyl alcohol. It comes out of solution, so we can see the clump DNA with our naked eye. So we'll place the tube into the centrifuge and click the lid to close it and turn it on. This time we will, after the sample spins in the centrifuge, the DNA sinks to the bottom of the tube. So once the liquid is removed and the DNA is allowed to dry, we can see redissolve in the solution of our choice. So we can store it in the freezer for many years, or we can move on to our next experiment. So we have just extracted DNA. So in the bottom of the tube, you can see the white precipitation. So there is the DNA. So that's all for DNA extraction. Thank you.